So, first of all, thank you so much for the invitation. The topic which I will be presenting today is the halotolerant plant growth promoting Rhizobacteria from Malaysian mangrove for crop resilience in the face of climate change and coastal flooding. Now, since the topic is mostly on the climate change and the effect of this climate change on the impact with the agricultural sector, mostly we are going to talk about, or the study what we have been doing revolves around those aspects. Now, before I start my presentation, I would like to state a quote from Dr. Guy McPherson, who once said, if you really think the environment is less important than economy, try holding your breath while you count your money. I know the quote would have brought a nice smile on your face, but the challenge is real. Climate change is happening now, and it's like devastating, and it's creating a serious impact on a lot of things which we can't even imagine. Wait. Now, the project which I'm going to present today actually started in Sibar in University of Malaya, where I was working as a postdoc and then research fellow for the last 6 years. If starting with the climate change and coastal flooding, rise in sea level is one of the most concerning and costly effect of climate change that impact sustainable development in the coastal areas. And Malaysia, basically, it presents 13% of the total land area within 5 kilometers of the coast, which is threatened by devastating impact of the rising sea level. Now you can just imagine with one thing that coastal flooding is very common in Malaysia, with Malaysia having the equatorial climate, constant high temperature and high relative humidity adds in with an annual average rainfall which causes flooding, which is actually approximately 2,420 mm. Now, with this coastal flooding in Malaysia, it is expected to rise, and the potential damage of these waves are expected to come at least once in the next 10 years. Now, you can just imagine the impact and how Malaysia cope up with all this kind of flooding. Now, it is also estimated that just 1 meter rise in the sea level is expected to cause the loss of 180,000 hectares of agricultural land. Now, with that includes 15 to 20% of the mangrove forest loss. Malaysia, basically, it has a very vast, nice forest stretch of mangroves, which actually crosses across 4,800 kilometers across the coast. Now, the image here, which shows the global pattern of the climate change in land salinization. If you see the downside of the Southeast Asia and South Asian countries, basically including India, Pakistan, Thailand, parts of Philippines, Malaysia, Indonesia, this whole area is actually one of the most vulnerable to high level in high rise in sea level, as well as the coastal flooding. Now, talking about the land salinization, we with this one only we can imagine that with this high sea rise levels also add in the land salinization. So when we talk about basically with the land salinization, it's not only NaCl what we work in the lab, but there are a number of salts. The salt, basically the primary salt constitute of sodium, calcium, magnesium, chloride, sulfate, and bicarbonate. These salts are very damaging to soil, and they percolate, they stay there, and they have their impact over the long run with each and every kind of productivity or the crop system, whatever is produced in that area. Now, most of the coastal areas within Malaysia is have very nice agricultural band which stretches along the coast as well as the sides. Now, with this land salinization, actually the groundwater level get disturbed. It has the unsaturated zone with the infiltration and evaporation coming in. The transpiration also adds in, which overall affects the productivity of the crop system. Now, with that, talking about, are these plants really able to tolerate any kind of this salt stress? Salt stress is basically categorized in the point of abiotic stress. Now, as we know that not every crop or not every plant is sustained or they are maintained to be tolerating any kind of abiotic stress. But with time and with going on time, and then with all this effect of climate change, all these plant and the microbial community which resides in the plant, they cope up with that condition and they come up with the stress induced plant what we know. So in that category, basically the salt stress causes two types of stress. One is the ionic as well as the osmotic stress. The ionic stress leads to disruption of ion homeostasis, which again disturbs the nutrient balance. And the osmotic stress leads to shortage of water with the closure and disturbs the closure of stomata. Now, to be very specific, this ionic stress causes nutrient deficiency, which means that they decrease the uptake of some of the prominent nutrient, which includes phosphorus, potassium, nitrogen, magnesium, and calcium. The ion toxicity later, it also causes increase in the uptake of the sodium ions. Now you can imagine with the increase in the uptake of sodium ion, they overall damage the photosynthetic machinery, transpiration, gaseous exchange, stomatal conductance, and also they lower the water potential of soil and leaves. Now with that overall impact it has, which causes, it goes to cell death as well as reduction in the growth, ultimately leading to the yield loss. Now, we have to think that not all plants are, as I already mentioned, not all plants are, how to say, tolerant or resistant to any kind of these abiotic stresses. But over the time, it happens or they modify themselves to make or to survive in that high salt stress conditions. Coming up, the primary plant which we have been studying in the lab is Musa acuminata, which is also commonly known as this is banana. Banana has a lot of benefit. The choice of this plant was because Malaysia is one of the biggest producer of banana. Most of these banana plantation are at the coastal areas, and the for this one, banana is actually a very good source of dietary starch. Now, the production of these banana is very important also for the farmers in the developing country because it adds in the value of their economy. Other than that, banana has a lot of important benefit, and then it adds on the it adds as a value added crop. Now, for the if I recall this one properly, this project with the it started with some of the preliminary analysis where we wanted to analyze what is the effect of this banana, what is the effect of the salt concentration over banana production. Now, if for banana particularly, it is a salt sensitive crop. It's not a salt tolerant crop. So we try to test different concentration of salt in the lab condition grown in the greenhouse. So you can see the figure 1 here at down. So the first one is controlled without any salt condition. Second one was moderately saline. Third one is severe, and the last one is extreme saline. The concentration of salinity, it was matched with the sea salt, which was tested for this analysis. With that, we were able to identify some of the morphological as well as biochemical changes. Particularly, it affected the root growth. As you can see in figure 2, the root growth was particularly hampered. The cell death was observed in the leaves, and we were able to do some of the biochemical analysis, including the total chlorophyll content as well as the carotenoid content. As you can see, the last graph, in the graph, the last point with the severe salinity 52.7, the overall rate of photosynthetic pigment, the changes in the photosynthetic pigment which was observed are shown here. Now, apart from that, we also tried to do some of the ROS content as well as some of the antioxidant enzyme. And then we were able to observe that how this, the one grown at the severe saline condition tried to cope up, and then how the level of ROS enzymes were related and antioxidant enzymes as shown in the figure. So we were able to do MDA content, proline, then some of the ROS content as well as the antioxidant enzymes. Now with that, uh, coming up with how these microbes support the plant and soil health. Now when we were doing this analysis, we tried to look for the as we saw the root section was already damaged a lot. We tried to look for all those rhizosphere related microbes. So particularly these plants are very smart or very active, you can say, because they have their own community, they have their own soil microbiome where they select each and every specific microbe which can go colonize and then can stay there and can help to replenish that salt or any kind of abiotic stress which has damaged the plants already. Now, in that area, if you consider plant rhizosphere microbes or rhizobacteria or plant growth promoting bacteria are one of the most neglected topic which is studied around the world. But it is very important to think that how these small microbes, they play a very prominent role in accelerating or in establishing the solid ecosystem around what we are living in. In the agriculture sector particularly, if we know these rhizosphere, the microbes, they can be divided into n number, particularly on the basis of their genera or their distribution. But what we wanted to focus was on the particularly on the rhizobacteria, which actually play a variety of role for nutrient cycle, then carbon sequestration, systemic resistance, the growth, and this stress condition analysis. Apart from that, we try to look that what is the pattern of these plants when they are introduced with these microbes, how the plant behave, whether they are able to cope up or whether these microbes can help to replenish those plant conditions, particularly in different level of salt condition. Now, why we chose and why we looked for the mangrove stretch, particularly if you see in this area which is marked in red, again the Southeast Asian region, here near the one which is shown in the dark, it contains all these countries which is Malaysia, Indonesia, and then Philippines, part of India. They have a very nice biodiverse mangrove forest. Now Malaysia actually has a lot of mangrove vegetation which is stretched across 4,800 kilometer across Malaysian Peninsula, Malaysia. Now, this one is a very much protected region since this is considered as one of the endangered site. And hence to work here and to get the sample collection, we required the prior permission from FDPN, which is Forest Department of Peninsular Malaysia, to carry out our study. What we did was, so if you see the location I have mentioned here, this region is come across the Klang port, which is between the Malaysia. It is called the Strait of Malacca. The region it is located in Carey Island, Selangor, and to your right is the mangrove plantation at the Carey Island. So as I mentioned, it has a very biodiverse stretch of mangroves which is present. We were able to collect soil from different sites with the tree sites. So the first one you see is one of the species named as Avenacia alba. The second one is Soneracia alba, and the third one is red mangrove, which is called Rhizosphere apiculata. Now, all these, the soil collection we did from the root region. We also collected some of the roots to isolate endophytes. Now, from here, what we did was the isolation was done, and then we wanted to also check what is how this after introduction of these bacteria or rhizomicrobes, what role they play or how they go and colonize to those particular root sections. For that, this one is one of our current project which is still in process. The estimation of root exudates. Now, when these plant in any kind of stress or in any condition, what they do is they try to release all these metabolites to attract those microbes and so that they can go and sit at their particular spaces and colonize. Those metabolites are called volatile organic compounds. The degree and the type of organic volatile organic compounds, they can vary across the plant, across the soil system, and they are really very important because at this point, the plant decide whether any pathogen will go and colonize or a beneficial microbe will go and colonize. So in this section looked very interesting, so that how they decide and how they determine that which one will go and colonize. So this is one of the part which we are still exploring. Then apart this one, the so how when they go and when they colonize, they have all these kind of activity which is going around, especially the one which can help for the nutrient replenishment includes nitrogen fixation, phosphorus solubilization. Siderophore production is for ion chelation, where the microbe they compete to join or to colonize the root system by utilizing the ion content. Then apart from that, they also help the root system or the plant to have external root formation with the production of this phytohormone, lytic enzyme, and some of the secondary metabolites. Other than that, they also participate in gene expression. So these are the things which we have explored, which I'll be talking about in some of my few of my upcoming slides. We were able to do some of the qualitative and some of the quantitative analysis. So just to mention that this study was already finished and the manuscript was submitted to microbiological research, which is still under revision. And hopefully we will be having the results soon from them. The first one shows, so as I already mentioned, we were able to work separately on rhizobacteria as well as endophytic bacteria. And we also wanted to check whether any of these rhizobacteria or endophytic bacteria, they colonize inside the plant, and if they have any role, particularly having the biphasic life cycle of these microbes to explore in that area as well. So the first figure which you see here is for the nitrogen fixation, how just the basic Jensen's media was used to check whether the nitrogen is, they are able to fix the nitrogen or not. The second one shows the qualitative analysis for the phosphate solubilization. Third one shows the siderophore production, you can see with the orange halozone. And the last figure for this slide is the quantitative estimation of phosphate solubilization. Now, overall, we were able to isolate approximately 80 to 85 microbes, including rhizobacteria as well as endophytic bacteria. When we screened them, we tried to do the initial screening first for all those microbes. So this one constituted, the qualitative analysis constituted all those 80 to 85 microbes. Quantitative estimation we did only with those who which has shown the growth in the high salt concentration. So we were able to select some of the isolate from that area. Other than that, then we went and we checked the growth. We tried to colonize these microbes in hydroponic media with particularly with the salt, particular salt concentration. The salt which was used to check here was 90 millimolar sea salt again. And the figure you can see, this figure is taken from SEM, which is scanning electron microscope. And you can see the pattern of colonization in all these roots when these roots were subjected for SEM analysis. So we were particularly able to observe that colonization actually happened for all these in all the treated plants. This figure here shows how the banana root, which is inoculated with the salt, and it was inoculated with the rhizobacteria and treated with salt, showed a higher, the length of the root was very nice, as well as the plant was looking very good. Now this one we compared from the first figure. What you see, A is with rhizobacteria and B is endophytic bacteria. And here also we were able to find one of the bacteria which was able to see very nice growth as well as the long root. This was the section of this root was then used for stereo microscopic observation, where at the left hand side on C, you can see the new root hair formation, and the D, which you see this side, is not showing any new root hair formation. Then this one with this one we confirmed that they are able to colonize the banana root system, and they are able to show the new root hairs, as well as they are able to replenish some of the nutrient which is basically known to degrade with the salt concentration or with the salt effect. Other than that, we were also able to check that one of these microbes, when we sequenced, it was the endophyte was one of the rhizobacteria was we sequenced also belong to the endophytic section. Now, in that part, we were able to find one of the information which is published by Dr. White in 2018, where he suggested that how these rhizomicrobiome not only they colonize on the outer surface, but they also have the tendency to enter from the root cap cells, which you can see in the section B. Now here they enter, and they inside they have this microbe plant entry zone, where the microbe become intracellular in the meristematic cell as the wall less protoplast. Now from here, if you see in the figure B, there are small sections, and this one is indicated by the black arrow in the root of the parenchyma cell near the root tip. Now from here they enter, and then they have all this nutrient cycle which is going on, where they have the nutrient extracted, and then they use it for the reactive oxygen production, and they pass on to the plant, and then they exit. So when they exit, on the figure C, you can see that how it's emerging, that the root hair tip of the millet seedling, the bacteria is exiting the surface, and then it goes out again in the rhizosphere. So that is one thing which was very interesting, although this was the first time we were able to see this, but it's already published in some of the manuscript. From here, we were also able to do the Evans blue staining for some of the banana, for the treated banana plantlets from the hydroponic section or from the hydroponic media, where we were able to see the first one you see is 1, 2, and 3, RB1, 2, 3, and 4. This is the one treated with Rhizobacteria. Then EB1, 2, and 3, this is the one treated with endophytic bacteria. The last one, 7, is the control. So you can properly see the cell death, which is visible from the image. Other than that, for this one, we also did some of the biochemical analysis, where we did the quantification of hydrogen peroxide and the measurement of MDA content. And we were able to see we had 4 set of experiment which contained the control plant, control with salt, our plant growth promoting bacteria, and the plant growth promoting bacteria with salt. Now, if you see the plant growth promoting bacteria with salt across all the section, it is showing very good. It showed nice pattern across all the tests here, what is shown in the image. We also did some of the antioxidant enzymes, where we were able to see the activity of catalase, superoxide dismutase, ascorbate peroxidase, and glutathione reductase in the leaves of the banana plantlet. Here also the one to be which was measurable and which showed very good result was the one, our rhizobacteria, which was inoculated again with the salt condition. So all these study made us to prepare this project for the industrial collaboration. And then we got a company who was interested in the project, and we have an industrial collaboration, and we are on the way to develop this using these microbes as a microbial biostimulant or consortia, which can be further used on the crops, not only in banana, but to other salt sensitive crop plants. Now, I'm going to show some of the preliminary analysis for the one which we have used the microbial consortia for salt treated banana plant. We did some of the morphological analysis where non-inoculated with salt, the consortia as well as consortia with salt, this was tested. So with the morphological analysis, we were able to check plant height, root length, number of leaves, relative water content, and plant biomass. And we were able to see that how the consortia, which is containing an endophyte as well as one rhizobacteria in the salt condition, was able to maintain and cope up with the one with the one compared to the non-inoculated with salt condition. Other than that, we also did the estimation of some of the photosynthetic pigment, which was chlorophyll A, chlorophyll B, carotenoids, some of the other tests which included proline and MDA. And with this also, you can see the consortia in the salt stress, it was able to perform approximately better or near that to the non-inoculated with salt condition that stressed plants. The comparison for all this study was done in 2 weeks and 4 weeks in lab condition, and the samples were extracted and they were taken for the analysis. Other than that is all the few of the antioxidant enzyme. The salt here used was the sea salt, which was even used for the hydroponic analysis. With this one, the test which were carried out for this one was catalase, superoxide dismutase, APX, peroxidase, and glutathione reductase. If you see for the antioxidant enzymes, particularly the test one with EB3 and RB3 with salt condition, we were able to see a very good pattern across all the antioxidant enzyme, what is shown in the image here. Now, coming to the last part of the result, what we have was, since we were able to analyze all these antioxidant and as well as the other related studies and biochemical study, we wanted to check whether any of the transcription factor genes or transcription factors, which is responsible particularly for salt stress condition, whether they have any effect over across these 2 weeks and 4 weeks of period during the inoculation with the salt and without salt. Now here you see there is the label here says the C is the consortia, NS is non-inoculated control with salt, CS is consortia with salt, and then this test was also done with 2 weeks and 4 weeks. We were able to test a number of WRKY transcription factors. And if you see the pattern of expression, we were able to see particularly WRKY 65, which was 65, which was found to be comparatively expressed more in comparison with 2 weeks. Other than that, 24 WRKY 145, 45, 28, 25. 23, 4, and 89. We were able to see all the selected WRKY. They were able to show the enhanced level of gene expression over a time period from 2 weeks to 4 weeks. Now, with this one, we are still doing some of the analysis to correlate the gene expression studies with how these microbes and the other antioxidant enzyme, how they work together, so that we can go ahead, we can present a PCA plot to show the correlation among this one, and then to form a basis that whether these rhizo as well as rhizobacteria as well as endophytic bacteria, how they are related, how they have an impact or they have a positive effect, particularly on the plant system which is in salt condition. This the study will form a basis which in the form of manuscript, as well as we are also on the way to prepare a consortia which can be used to analyze the or to actually enhance the or provide the nutritional value and enhance the salt condition or the abiotic stress condition, particularly in other plant species. So that's all from me. This is our research group. The research assistant, Abdul, Dr. Kawi, he is a co-researcher, Dr. Purabi, she is the senior lecturer, Dr. Najia, she is the research officer. She has helped with all our paperwork and other related matter. Dr. Purabi and Dr. Kawi has been helping with the analysis and the plan of the experiment as well as the manuscript. Our principal investigator, Prof. Jenny, and this is me. The project is funded by within University Malaya at UMCIC. We have a collaboration with Kimagri, the company which is helping and then also analyzing some of the plant experiment at their field in Cameroon Highland, and Farmers Biofertilizers and Organics in India. They are also one of our partners. So I would like to thank the SATU team for organizing this one. Other than that, this is the manuscript which we have, some of the related manuscript rather, which we have submitted as well as published in the last few years, particularly related with the sea salt and the transcription factor in banana. This one is my favorite slide where you can see us collecting sample from the mangrove. This is Abdul, Prof. Jenny, me and Puravi. And then some take home message, the facts about mangroves. I have just attached so that you'll know that how important they are and how they can help us in the climate change. I think that's all from me. Thank you so much for your time.
Thank you very much, Dr. Pooja. And so I think now we'll move on to question and answer session. So I think at the bottom of your screen, you should be able to see a raised hand, like a little hand symbol. I know some people have been waving. So if you have a
Okay.
question, you can click on that and I will try to find you in the participant
So
list
I'll
and
just
invite you.
stop
Yeah.
sharing first, right?
I think you can keep sharing in case people want to
Oh,
refer to the slides.
yeah, but then I cannot see the main.
We will call out the questions for you. I think Charlotte will help me with that. Or you can, if you don't want to use the microphone, you could type into the chat box and we can read out your questions for you. So
Okay.
either method will work. Okay, I'm looking to see any raised hands.
Yeah, please feel free to ask. If I'm not able to answer, I have my team here who can help to answer.
From our experience, the audience usually needs some time to,
Okay.
some time before they ask a question, probably because they need to digest so that they can ask a question.
Okay. Yeah, sure.
So I think one interesting observation I would make is I think all of the countries that we have participants from have quite considerable mangrove and mangrove resources, and so this is something that is also under threat. I think it's one of the ecosystems that has to be protected and can be a tremendous resource. It's not just for finding microbes. Actually, they are quite a barrier against tsunamis and even coastal erosion. So they're very, very important ecosystems. And that was one of the reasons we wanted to do, help to show their value through some of our research. In Malaysia, they are actually protected, but still we see them being lost sometimes to development because, coastal regions are obviously in high demand for many activities. Yeah.
So is there any plan to expand the scope of your research to other mangroves in other countries?
So that was one of the things we wanted to do. We wanted to maybe do some comparisons. If anyone else here is actually sampling microbes from their mangroves, we'd be really interested to see how similar the microbe species are. Particularly if you have similar, I think many of you would have similar tree species to the ones that we have sampled, or even if they're different for different tree species as well. So, it could be quite a big study if we got lots of people involved looking and comparing at what we all have. I think that would be really interesting.
So
Yeah,
any
to just add to Prof. Jenny, actually what we are looking is only one part of the work. It's only salinity what we are looking. But they do have a lot more, if you consider the microbiome. So there is one project which I'm currently doing since I have left SIBAR, but I'm still working with SIBAR. One of the project which I'm doing is the analysis of metagenome in mangroves. So the metagenome will then, it has n number of things which is going around, specifically the seaweeds which come in and then they get attached to those mangroves areas. The seaweed, particularly the microbes in the seaweeds, the endophytes in the seaweeds, fungus and all, they have a lot more benefit rather than only the soil bacteria. So that is, all those areas should be protected properly and they have enormous benefit not only for one section, but other abiotic stress or other kind of plant related or agricultural sector studies. So that is one, I think,
Yeah,
since
Dr. Pooja, we have a question in the text
okay.
box from Trin.
Okay.
Trin is asking, how did plants recognize VOCs to know the microorganism is either a pathogen or can benefit plant growth?
Okay, thank you for your question. So I'm going to give a very small example. and then I will leave you to think and then study. One thing is, so for example, if we are looking for particularly, for example, a banana plant or any other plant, in any stress condition, now they are in need of all those plant, all those rhizobacteria or all those microbes which are in that microbial community to, for their nutrient deficiency and all those things. Now, in that condition, if you see, for example, there is iron which is present in the soil in different form, obviously not like iron, but in different form, mixed form. These microbes, the beneficial microbes as well as the pathogenic microbes, they both compete to use that iron for their growth. When they grow, they try to go and attach to that section. So during the attachment, the plant already start releasing all these root exudate, which comes as a metabolites. Only those microbes will go and join, and then they will utilize those metabolites, which can utilize the iron as the source of their energy. Now, in that competition, so you can just imagine that it's even a part, it's like a competition. Other than that, for example, if you consider other scenario, if the plant is wounded or if the plant is even near the damage or stressed, it may not be able to have all these organic metabolites releasing so that plant rhizomicrobiome can utilize. In that condition, the pathogen which are present in the microbial community, they will win. So they will compete, they will win because the plant is already stressed. The plant may not be able to send any signal to avoid those pathogens, and these pathogen will go and join. So that is how it works. So for a healthy plant, a healthy volatile organic compound and a healthy root exudate system, it is very important that these plant environment, the soil and other things, they have to be very healthy. I think I have tried to answer a bit.
Yeah, so I will just interject that because I know for Malaysian audience, they will talk about ferrum rather than iron, just in case anyone is not
Oh,
clear.
okay.
Yeah,
Okay.
so ferrum, iron, we're talking about that. Yeah.
Is that have I answered? I cannot see who.
Trin, is that what you wanted to know? Sorry, I don't know if you're Mr. Trin or Miss Trin, Dr. Trin. Okay, yeah, say
He
thank
said
you.
thank you.
Yeah,
Yeah.
thank you. Yeah.
Okay, thanks. Yeah.
If there are other, very soon we have another question from
Okay. Are the developed
Emma
to
Calestra.
suicide risk? I can see, but what is, sorry, I cannot see the question.
Oh, okay,
Okay,
I can read it. So
okay.
are they developed to be used for seaside rehabilitation for polluted waters? So I guess this is about the microbes? Yeah.
Microbes or plants?
I guess he's asking about, are you asking about the microbes or the plants? The microbes.
Microbes.
Oh, microbes. Okay. I'll try to explain this one. What happens is that if you imagine this microbial community, they are actually not the one which are like you can identify some random and then you can state that this is the one who which should be working. Rather, it works the other way around. Now, in any environmental condition, these microbes are settled. They are living peacefully, eating, dancing, everything. Now, suddenly a stress condition comes, suddenly a salt condition comes, suddenly something comes. These microbes need to survive. They need the carbon source anyhow. So, slowly and steadily, they develop themselves to adapt to that condition. And they go from from a scale to 0 to the extreme one since they need to survive. So particularly talking about the adaptation, they have adapted themselves well to suit in this environmental condition, but they also have retained to have all these properties what we have seen. Now, particularly to answer that why they have retained this property, that may be a bit difficult because the proper profiling or screening has to be done from where it started. But with my understanding and what research I have been doing, I can just tell that slowly they have adapted themselves, and so they are also making with all these activity, they are making the plant to adapt to this high salt condition. And so only we are not discussing the transgenic banana today, And we are discussing the other side, that what can be the alternate, which is how we can use these microbes to make banana adapt that salt stress condition. Yeah, so that's from me. I hope I have answered. Yeah.
Is that a good answer, or you want to ask something further? It says thank you.
Thank
Okay.
you.
Okay. Okay. Thanks. Thanks.
Yeah. I'm trying to look for any more questions.
Or raised hands.
I don't see any raised hands.
Our audience are very shy usually.
They're
They
very
tend to type the question rather than speaking it out themselves.
fine. I'm happy to read them out if you want to type them. Yeah.
Okay.
We have a new question.
Okay. So Marika is asking, there's usually concern in introducing foreign organism for solving ecological problems. They may become invasive species. Is there a similar concern to using microbe from the mangrove to solve issues in farmland?
Okay.
So could it become an invasive species?
Okay. Okay. If you ask me particularly, yes, there is a concern. And so only, I think, with my understanding, whatever we have been looking for is first doing a very invasive and nice initial screening where we can have a proper identification, sequencing, genome analysis, and how they can cope up. Because you have to think that we are not taking these microbes from any different this one. They are the soil microbes which are attached to roots of a plant which is in an extreme environment, and we are trying to match those stress condition in the agricultural soil, which is again showing the kind of salinization slowly, gradually over a time. So yes, it has the chances of becoming invasive species, I agree. But the point is, what about all those advanced techniques what we have been using for screening and then across the genome? So I think if we take step by step rather than just going and throwing anything in the soil, the steps for the initial screening here is important. But I would like to ask Prof. Jenny if she can add something more on that.
Okay, I think it's an excellent question, and it's one that maybe would require more someone with ecology background. So I will not claim to have an ecology background. But one of the things I think, for all of the mangrove rehabilitation, the same questions come too, and people are replanting to make sure you are replanting with the species that, and even the genetic materials that are native in the area. I think as far as possible, you shouldn't be moving things too far. So if possible, and you were talking about agricultural areas adjacent to where the mangrove species are, it's more or less disturbing than moving them, say, to a different country or to a different region. So I think those ecological aspects certainly have to be considered for any of this. We know how unnatural and unecological agriculture is in general. So I think there's a strong move to try to be much more environmentally friendly and sustainable for the long term. So all of these aspects are very important to consider. So thank you for that question. I think we just have some comments, not questions there. Okay, thank you. Thank you for your comments. Okay, I don't see any new questions. Charlotte, I cannot hear you.
Yeah, sorry, I muted myself. So, Professor Jennifer, do you have anything to add based on Dr. Singh's sharing?
So just to thank her for sharing what we're doing as a group, and to mention, as she did, that this is very much ongoing research, and we are
Yes.
interested if people want to contact us and explore potential collaborations. We're continuing to write proposals and try to move our research forwards, as I'm sure everyone is. And
Yeah, please.
I just want to say I thank you for the opportunity for us to present our work. I really appreciate that. Thank you.
Thank you very much for sharing your work. I can see there's lots of effort spent on the work. And then one more suggestion is that I suggest that next year you can use this research to run a bigger team. Maybe you can join our JRS next year so that you can match with scholars from other countries and then to enlarge the scope of the research.
Yeah, we would certainly like to do that. Thank you.
Yes.
Yeah.
If we do not have any other, is there any other questions from the audience? If not, I think that would be the end of our section. So for the audience, if you can think of any question you would like to ask Professor Jennifer or Dr. Singh afterwards, please send us a question at the link satu@ncku.edu.tw, which is the email address that you received the WebEx link that sent you the WebEx link. So I think that would be all. Thank you very much for this very informative section. And thank you very much for sharing, Professor Jennifer. Thank you for the sharing, Dr. Singh. And I have a very informative afternoon. It's very enjoyable. Yeah. Thank you very much.
Thank you. Thank you so much. Thank you for the invitation.